Hello everyone, welcome back. So this week we're going to be covering exercise 10, which is kingdoms, bacteria, protista, and fungi. Today I'm only going to be covering kingdoms, bacteria, and then I'll post another video that covers protista and another one that will cover fungi in the future. So I'm going to go ahead and pull up the PowerPoint real quick. So hopefully everyone is seeing my PowerPoint on the screen. Okay. Okay, so like I said, we are going to begin looking at all the different kingdoms that make up life on Earth. And we're really going to do this for the following few weeks. So as we go through this, really try to pick out key features of each kingdom, what makes them unique from another kingdom, and I will tell you specifically what you want to know. For example, when we cover the kingdom bacteria, you're really going to want to focus on how to distinguish different bacteria, and this is going to be based off of their shape. You can also distinguish them based on color using a certain type of stain that I will discuss. And then bacteria can also be found as both anaerobic or aerobic organisms. So just things to kind of think of as we go through these next few lectures. And I believe this is going to be the next few weeks. I don't know. I think Kingdom Animalia, so the animal kingdom, will be the last one we cover, and that's going to be exercise 12. So we're going to begin looking at these kingdoms. We're going to begin by looking at microorganisms. And microorganisms can come both from prokaryotes and eukaryotes. Okay. So the first thing I want to do is introduce the kingdom systems that we use. So we used to use Whittaker's five kingdom system, and this system divided prokaryotes. They classified them as Monera. Then you had your protists, your plants, animals, and fungi. So there was five different kingdoms here. And this was your prokaryotes. Everything from the protists up were your eukaryotes. Since then, this has been reworked, and in, I believe it was 1990, the prokaryotes were split into two different domains. So now we refer to it as the three domains of life. And these prokaryotes were split into bacteria and archaea. And then you have your eukarya or eukaryotes over here, and your eukaryotes consist of plants, animals, fungi, and protists. Protists are really this catch-all. So if it's not an animal, if it's not a plant, and it's not a fungi, it's a protist. And we will discuss protists later on, but they can either be animal-like or plant-like protist. Okay, so what else? Oh, and the way these are based, the way these were grouped into these three domains was based off of their RNA profiles. So they realized that Monera, the RNA profiles for bacteria and archaea were so different that they didn't belong to this Monera group. They shouldn't be grouped as one, so they got split into two. Okay, so like I said, we're going to begin with microorganisms, and these are essentially microscopic organisms that can exist as either unicellular, multicellular, or colony of cell clusters. They are widespread in nature, and they can be both beneficial to life as well as harmful, and I will try to discuss how archaea and bacteria, for example, can be both beneficial as well as harmful. And like I said, microorganisms exist in both prokaryotes and eukaryotes. A lot of times we associate microorganisms with bacteria and even archaeas, but they do exist in eukaryotes as well. So we're going to begin with prokaryotes. And in this lab, we're actually only going to be discussing bacteria, but I still want to introduce the archaea. So archaea, essentially, live in very extreme conditions, and they are classified as methanogens, thermophils, or halophils. So they fill meaning loving. So either salt-loving organisms. They can survive in either extreme weather, whether that's heat or extreme cold, and then they produce methane. Bacteria, they are essentially everywhere. They could be either aerobic or anaerobic. They can be producers as well as consumers. So as I said, archaea, we're not going to really discuss them here, but just want you to know that they are extremophils. So they are an organism that hasn't evolved to thrive under chemical or physical extremes. So we have methanogens, and this is showing how we take advantage of these microorganisms in water treatment. So methanogens can produce methane, and they are really good at breaking down sludge found in sewage systems. So a lot of countries take advantage of these organisms in water treatments. You have halophils, so these are salt-loving organisms. And here I have an image of the San Francisco Bay salt ponds. So these were essentially used as salt evaporation ponds since the gold rush, obviously to preserve food. Salt is great at preserving foods. So that is why these banks were taken advantage of and used for those purposes. They are now part of a restoration project, so we're trying to get it back to its natural wetlands. And this is to provide food control. This is to reduce pollution and to expand the habitat for wildlife. There's a lot of benefits in trying to restore the natural wetlands of these salt ponds found in the San Francisco Bay Area. And this is an image from an airplane. So as you come in and land into San Francisco, you can actually see all these bright colors. And those are all those microorganisms, and just like the way they interact with these salts, they produce these very bright, vibrant colors. And then you have the thermophils. Like I said, they live in extreme conditions. It could either be extreme heat or extreme cold. So this is just a general structure of prokaryotes. So one, they are significantly smaller than eukaryotes, and there's actually an image showing the two side by side, so you can really get a visual of how much smaller prokaryotes are than eukaryotes. If you remember back to our previous lecture, prokaryotes do not have membrane-bound organelles. They have circular DNA, so their DNA is just like one chromosome. It's found in this loose spaghetti-like pattern here, and it's referred to as the nucleoid. So remember, in eukaryotes, our DNA is tightly packaged and wound around those histone proteins. We're really trying to package as much DNA into the chromosomes that make up our genetic DNA. They also have ribosomes. So as you can see, they have ribosomes. The ribosomes are used for protein synthesis in prokaryotes, just like as we have seen in eukaryotes. The difference is that prokaryotes use a 70S ribosome, whereas eukaryotes use an 80S ribosome. Some produce endospores, and this is for survival purposes. So the best way I can talk about this is if you work in a laboratory, you have to sterilize your biohazard waste. And when you sterilize it, you will put an endospore on your waste to ensure that it is able to that it is at high temperatures, it is actually sanitized. And that's really how I use endospores in the laboratory. But bacteria have evolved these purposes for survival purposes. They also have flagella. So the flagella is shown here, and this is used for motility. And it can have one flagella. It can have multiple flagella coming from this end. It could have flagella coming from all over the the capsule. It could have just one flagella here at this end and one flagella here. So that can really vary depending on the type of prokaryote you're looking at. Most will have a cell wall, which is shown here. The cell wall, so you have your plasma membrane, and then you have the cell wall, and then you have the capsule. So the plasma membrane is yellow, cell wall is this blue color, the capsule is this green color. So in bacteria, you're also going to have this layer called the peptoglycan layer, and we're going to talk more about this when we talk about the Gram staining that we use to differentiate between different groups of bacteria. But essentially, this is a sugar protein polymer. Archaea do not have this peptoglycan layer. Okay, so moving on to bacteria, they are very abundant and very diverse. They have been here for a very long time. We already went over some of the characteristics of them. They have circular DNA. Some of them will have extra small DNA in the form of plasmids. Their cell wall is composed of peptoglycan. They reproduce by binary fission, and they show great diversity in metabolism and habitat. So they can thrive in both anaerobic and aerobic environments. It really depends on the bacteria that you are dealing with. And there are certain ways we can classify bacteria. One of the ways we can classify bacteria is based on shape. So bacteria come in 3 different shapes. So you have a rod-like shaped bacteria that we refer to as bacillus. We have the spear-shaped bacteria, which we refer to as coccus. And we have a spiral-shaped bacteria, which we refer to as, I actually refer to this as spirillum, or spirillus is what it's shown here. So I just want to give you some examples of each group of these. So an example of a bacillus or rod-like bacteria is anthrax. So anthrax is a serious infectious disease caused by gram-positive rod-shaped bacteria. So as you can see, it has this rod shape. You can also see from this image that these bacteria stain purple, and that's why we say gram-positive. I will talk about this in another slide more. And these rod-shaped bacteria are known as Bacillus anthracis, Bacillus anthracis. And many times the shape of the bacteria is used in the name. However, that is not always the case. I'm also giving you guys examples of bacteria that are considered pathogens or infectious diseases, but not all bacteria are bad. We actually have a lot of bacteria that live in the gut flora and that are essential to digesting different meals and, really using the nutrients from our meals for other purposes. So not all bacteria is bad. We actually want some level of bacteria and some types of bacteria in order to fight off infections. Okay, so another example of a bacteria will be strep throat, which is an infection in the throat and tonsils caused by the bacteria called group A Streptococcus or Streptococcus pyogenes. So this same bacteria is also responsible for a variety of diseases. So depending if it gets into the blood and it becomes systemic, it can lead to flesh-eating bacteria. So if you look at these images, you will see that these have circle-like shapes. They're also purple, so they are gram-positive bacteria because they stain purple. And I just want to point out that these circle-like shapes, they can be found just like one here, one here, one here sporadically. So just by itself, they can form these chains as shown here. They can also be found in groups of 2, like just 2 right next to each other. Those are referred to as diplococcus, or they can be formed these tetrates, which would be 1, 2, 3, four of them grouped together. So here is an example of, oh, and I just want to point out that the shape of the bacteria is in the name for this specific bacteria as well. So Streptococcus, it tells you what shape it has. Here's an example of something that the name doesn't quite tell you exactly what shape it is. So Neisseria gonorrhea, so we know this as a type of STD, also known as gonococcus, singular gonococci, plural, is a species of gram-negative diplococci bacteria. So this is that bacteria that I was telling you that comes in groups of 2. So it is referred to as diplococci bacteria. And as you can see from this image, it stains pink. So it is a gram-negative bacteria. So syphilis is a bacterium called Treponema pallidum, and as you can see, it has this spiral shape. So this is a Spirillum bacteria, and it's I believe it's gram negative. I didn't use an image where it's stained with gram positive or it's stained with the gram staining, but I believe it's gram negative if I recall correctly. So I told you guys that we can classify bacteria based on shape. These are the 3 basic shapes. I want you guys to feel very comfortable with each of these shapes. You can also classify bacteria according to how they react to the gram stain. So bacteria can either be gram positive or gram negative. When it is gram positive, it's going to produce this purple color. When it is gram negative, it's going to produce this pink color. So why does one become purple and why does one become pink? It has to do with this peptoglycan layer. So bacteria that is considered gram positive has their plasma membrane and then a huge chunk of this peptoglycan layer. The gram stain is or the reagent we use for gram staining is called crystal violet. Crystal violet is going to attach to this peptoglycan layer, giving it that purple color. Well, when we look at the cell wall of gram-negative bacteria, you have the plasma membrane, you have the peptoglycan layer, and then you have this outer membrane, which is called the capsule. And then on top of that, you have LPS, lipopolysaccharide, which is an endotoxin on top of this outer membrane. So it is much more difficult for this dye, this crystal violet, to get to this peptoglycan layer in this gram-negative bacteria compared to here. There's nothing protecting this peptoglycan layer here. Here we have this capsule and the LPS protecting the peptoglycan layer. So when you add crystal violet here and you add crystal violet here, crystal violet is going to attach to the peptoglycan layer, giving it a purple appearance. Here it's just going to come right off. The next thing you do is add iodine. You do an alcohol wash, which is the decolorization, and then you apply this pink stain. Safranin, it's a counter stain. When you add it to the gram-positive bacteria, it doesn't do anything because it's already stained purple, so it doesn't matter. But when you add it to the gram-negative bacteria, it now has this pink appearance. So that is why gram-negative bacteria will appear pink when stained with this crystal violet gram stain, and why gram-positive bacteria will stain purple. So I want you guys to go ahead and pause here for activity 1, shapes and characteristics of bacteria, and I want you to look at these different slides. So if we were in lab, you would look at different slides that are already prepared for you under the microscope, and you would look at these slides and first identify which one has the coccus-like shape, bacillus-like shape, spirillum shape, and then you'll draw here what you see. You'll identify what color it is, and you will tell me whether it is gram-positive or gram-negative based on whether it is purple, stained purple, or pink. So for activity 2, you would have taken a toothpick and scraped your teeth with it, and then you would have prepared a slide. You would have stained that slide with methylene blue. and then you will have looked at your prepared slide under the microscope. You would have seen something like this. So dental plaque has a lot of bacteria in it, and it's mainly composed of Streptococcus bacteria. So coccus, remember, that's going to be that circular-like shape. And when you look under the microscope, you will want to draw what you see. So here you see two very different cell types. You should see prokaryotic cells, which are these very small round cells. They're very dark, stained very dark. And then you should see these other eukaryotic cells. These are most likely squamous epithelial cells from the cheek lining of your mouth. As you can see, these squamous epithelial cells have this dark stain in the center. This is the nucleus. Remember, eukaryotes have nucleus. So that is what is staining this deep blue color here. Okay, so that would be the next activity you would do in your lab manual. It's part of activity 1 as well. So now moving on to activity 2. So cyanobacteria, or blue-green algae, are autotrophic photosynthetic bacteria. They are typically found in aquatic environments. And I have different images here showing various cyanobacteria that you can typically find in pond-like water. So this is they're referred to as cyanobacteria blooms. When you see this in the lake, this is cyanobacteria, essentially. This is photosynthetic bacteria. Let's see. Sorry, I'm just pulling up my notes to make sure I talk about everything I need to talk about here. Okay, so cyanobacteria are extremely old. They are about 2.5 billion years old, or they've been around for 2.5 billion years. And cyanobacteria are really neat because they brought something new to the planet. So when the planet first began, it had certain gases available. It had CO2 available from volcano, and it also had water available. However, it had no oxygen. So when life first began on Earth, we had gases available such as CO2 from volcanoes, and we had water available, but we didn't have oxygen. So cyanobacteria are very special because they brought this missing element that gave rise to organisms like you and I that require oxygen. And this is referred to as the Great Oxidation Event. It is also sometimes referred to as the Great Oxygenation Event. So Archaea, so we already talked about Archaea, and Archean Earth was anoxic. It did not have bacteria, and most of those organisms are anaerobic. But something happened at some point where cyanobacteria used light to split water into hydrogen and oxygen. And I'm going to come back to this. But something happened, and it was able to use light to split water into hydrogen and oxygen. And this led to oxygen being released into the environment. So when oxygen was released into the environment, it did a few things. It produced the ozone layer, which shields us from dangerous UV wavelengths of light. And then it also made aerobic metabolism possible. So it was able to provide organisms with more energy than anaerobic metabolism. So as we already discussed earlier, aerobic metabolism is able to generate a lot of energy when we think back to cellular respiration versus anaerobic metabolism. So this allowed the capacity for more complex life. So the being able to have aerobic organ or being able to have aerobic organisms gave rise to a more complex life on Earth, which would be you and I. And the other thing that cyanobacteria really did was that it is the origin of plants. So this gave rise to plants. Cyanobacteria have chloroplasts. I'm sorry. Plants have chloroplasts, and the chloroplasts found in plants actually came from cyanobacteria. So if you remember back to when we talked about mitochondria, I told you that at some point in evolution, a eukaryote consumed a mitochondria and found that it was beneficial to have this mitochondria living in it. So then it decided to keep it. Well, the same thing essentially happened with plants and cyanobacteria. At some point in evolution, um, a eukaryote consumed cyanobacteria. This provided shelter to the cyanobacteria, and it provided this eukaryote or plant with chloroplast. And then it decided, you know what, it's really nice having this chloroplast. I'm going to keep this. And then it decided to keep that. And this is an example of, this is what we refer to as endosymbiosis. So these are the two big things cyanobacteria contributed to life on Earth as we know it. There's a lot of questions being asked as to what led to cyanobacteria all of a sudden being able to produce oxygen from water. Like, how was it all of a sudden able to use light to split water into hydrogen and oxygen? Like, why couldn't it do that earlier? Why did it take this much time? And it's really difficult to answer these questions. There's a lot of theories out there. Some people say, well, it took time to develop the trait to bump oxygen production even more. Other people say that it was gradually producing this oxygen, and then eventually it hit this point where it produced a lot, where it was able to sustain aerobic metabolism. We're not quite sure. There's a lot of theories out there that you can read about. But more on cyanobacteria. Cyanobacteria blooms are very dangerous. We talked about the good things cyanobacteria gave us. It gave us oxygen and it gave us plants. The bad stuff about it is it builds up in lakes a lot of times in these cyanobacteria blooms, and it's very toxic to both humans and animals. There's a lot of warnings in certain areas, such as Lake Erie, for example, gets a lot of warnings about cyanobacteria blooms during the springtime. And yeah, that's about it on that. And cyanobacteria does produce various cyanotoxins. There's not really a lot of resources available to combat cyanotoxins, and not many labs test for them. So you really are just told to reach out to poison control. But that's it on cyanobacteria. Now we're going to move on to activity 3. Oh, so for your lab manual for activity 2, you would draw what you see in the space provided. So you have an image here of oscillatoria. Oh, actually, that's not even oscillatoria. That's something else. So I think that was a mistake in your lab manual, because oscillatoria looks very different. I'm looking at the old lab manual, so they might have updated it for you. But go ahead and draw an image of cyanobacteria in your lab manual in the space provided. And I have some examples here, but if you want to look up oscillatoria on Google, you can just go ahead and draw that. So now we're going to move on to activity 3. And many of you guys are probably familiar with antibiotics. And antibiotics were first discovered by this scientist shown here. His name is Alexander Fleming. And in 1928, he made a chance discovery that revolutionized um, bacterial infections. So he discovered this antibiotic that we call penicillin, and it was actually just by accident. He had a bacterial isolate that he was trying to find a remedy for, find some kind of therapeutic treatment for, and he couldn't find anything. And then one day he left his agar plate out by a window that had a bacterial culture on it, and it started to grow mold. Um, and he saw that the mold was actually killing the bacterial organism he had on his agar plate. So that is how he was able to discover penicillin. So certain molds produce a compound that inhibits the growth of bacteria. And he reached out to two other scientists where they were able to isolate this compound, and then they called it penicillin. So penicillin's mechanism of action is it acts by inhibiting the biosynthesis of the cell wall. So the cell wall is synthesized in 3 steps, and it inhibits the last step of cell wall synthesis of bacteria, which is cross-linking of peptoglycan. So remember we talked about peptoglycan earlier, and that is the mechanism of action of penicillin. You do not need to know that. So here are some the structures of a gram-positive bacteria. So if you remember, it has this nice thick peptoglycan structure, and then the cell membrane underneath it, and then these green objects are just transmembrane proteins. And if you were to add that crystal violet dye here, this whole peptoglycan would turn purple. So this is referred to as a gram-positive bacteria. Here is an example of our gram-negative bacteria. So this gram-negative bacteria has your cell membrane down here with your membrane proteins shown here in green. This brown thin layer in the middle is the peptoglycan layer. Remember, that's your sugar protein polymer that is stained using the crystal violet gram stain. And then you have this outer membrane layer protecting that peptoglycan. This outer membrane layer is also referred to as a capsule. And then you have this LPS, lipopolysaccharides, at the top. These are those endotoxins that are also protecting these other layers. So when you add the crystal violet dye to this bacteria with this cell membrane, it will not stain because it can't reach the peptoglycan layer. Well, if you think about antibiotics, you treat gram-positive bacteria very differently than you would treat gram-negative bacteria. So antibiotics mechanism of action target things that are unique to bacteria. So we want to make sure that antibiotics target something unique to bacteria and not something that is found in human cells, because then we're going to be killing our own cells. So that's the first step. Antibiotics are going to target something unique to bacteria, something that is not found in eukaryotic cells. So then once your doctor identifies whether you are dealing with a gram-positive bacteria or a gram-negative bacteria, so is their patient dealing with, I guess, syphilis, or is there a patient dealing with streptococcus? Once they make that determination, they will treat their patient very differently, because antibiotics typically, they don't always act on the cell wall, but our older antibiotics do act on the cell wall. So they act on 3 different steps of cell wall synthesis. So step 1, step 2, or step 3. Penicillin acts on step 3, which is the cross-linking of peptoglycan. But essentially, these antibiotics are going to act very differently for gram-positive versus gram-negative bacteria. So this leads us to activity 3, which is the antibacterial property of household items. So if we were in lab, we would have an agar plate that is streaked or inoculated with bacteria. You would inoculate one plate with the gram-positive bacteria and another plate with the gram-negative bacteria. So here the gram-positive bacteria you're going to use is Staphylococcus aureus, and then the gram negative would be E. coli. And then you would take these paper discs, and soak them into different cleaners. So you would use one disc soaked in Bactine, and you would place it in quadrant A. You would take one disc, soak it with Dawn, which is the dish soap, put it in quadrant B. You would take one disc and soak it in Lysol. So you're going to saturate these discs essentially in a cleaner. So here, Lysol, put in quadrant C. And then you take one disc and saturate it with Clean and Clear, which is a face wash that is used for acne-prone skin. So acne is due to bacteria. And then you would put that in quadrant D. And then you would see what happens to your plates. So remember, we're using two different bacteria. We're using one that has a gram-positive and one that has a gram-negative cell membrane. So our gram-positive bacteria is going to have this type of cell membrane. Our gram-negative is going to have this type of cell membrane. And I just want to point out that these agents are all antibacterial agents. You would not, some of these might have antiviral properties as well, but all of these agents, A, B, C, and D, have antibacterial properties, not antiviral properties. Bacteria and viruses are completely different. You wouldn't treat a virus with antibiotics. You may use antibiotics in virus infections if that virus caused a bacterial infection. But if the virus did not cause a bacterial infection, you don't use antibiotics. You just use antivirals. So this is our plate that was inoculated with E. coli, and this is a plate that was inoculated with Staphylococcus. And as you can see, in quadrant A, B, C, and D. So here's the legend: A, B, C, and D. It seems that Dawn and Clean and Clear are effective against E. coli. When we look at our gram-positive bacteria, we see that all of them are effective against our gram-positive bacteria. So you would answer the following questions. Which type of organism is inhibited more? So if we look at these two plates, we will see that our gram-positive bacteria is inhibited more. It was inhibited by all 4 agents, whereas our gram-negative bacteria was only inhibited by the antibacterial properties of B and D. So which chemical agent worked the best? So in my opinion, B and D seem to work pretty well here, and B and D seem to work the best over here as well. Which chemical agent was the least effective? So overall, it seems like A was the least effective in both plates. And this cloudy area is where the bacteria is. This clear area is where the bacteria is dying. So it's this antibacterial household cleaning item is killing the bacteria there. Okay, so moving on to activity 4. So if we were in lab last week, you would have received an agar plate, and you would have put your thumb or a finger in quadrant 1. Another classmate would put it in quadrant 2, another classmate in quadrant 3, and another one in quadrant 4. And essentially the same thing would happen here. Or you could use one plate for one student. I think that's actually what they do. So if we were in class last week, you would have received an agar plate, and you would have put a fingerprint into each quadrant, 1, 2, 3, and 4. Another student would put it into a different plate, 1, 2, 3, and 4. You would give it a week for the bacteria to grow. You would come back the following week and look at the bacteria. And you would just observe these plates and ask yourself different questions. You would ask yourself, can you see different types of bacteria? Can you see different types of fungi? how many colors of bacteria and fungi do you see in total? And this is just a joke here showing if an 8-year-old put their handprint on an agar plate, you would see all sorts of bacteria and fungi. So I am showing here a HE agar plate. So if someone was suspected to have, if someone had stomach pains but didn't know whether it was due to Salmonella or Shigella, you would collect their stool and streak it on an HE agar plate. So an HE agar plate has different nutrients, and depending on the type of bacteria it is, it's going to react differently on the plate. So essentially in quadrant 1, you streak it very heavy, which is shown here. In quadrant 2, you go a little lighter. In quadrant 3, even lighter. And then in quadrant 4, it's extremely light so that you get these isolated colonies. So if your patient had Salmonella, the bacteria would turn black on the HE agar plate. If your patient had Shigella, it would stay this kind of silver neutral color on an HE agar plate. If your patient had, I believe this is CRE resistant bacteria, it might be E. coli, it turns this fluorescent orange on an HE agar plate. This is the same plate, the HE agar plate, and it turns this fluorescent orange. So you use different type of agar plates that have different nutrients to determine what type of bacteria you're dealing with. So you, when you inoculate these plates with the bacteria, you want to look at size, you want to look at color, you want to look at shape. I put size on here twice, sorry. Texture, margin, and elevation. So size, you can see these are very small. Some of these look much larger over here compared to the Salmonella. Color, this one's black. This is kind of like a neutral color. This is a fluorescent orange. Shape, sometimes they have these irregular edges. Sometimes they're very round, as shown here. Texture, you want to look at the texture. As you can see here, sometimes they look more wet and kind of like gooey. Sometimes they look super dry. You want to look at the margin. As you can see, the margin here is irregular, whereas the margin here is pretty defined. And then you want to look at elevation. Some of these have very kind of like 3D-like elevations. Some of them are extremely flat. These seem to have like these kind of like beads. They also have some elevation to them, but some are completely flat. So I have some more examples here. So this is on a blood agar plate. So as you can see, different bacteria will grow very differently on a blood agar plate. So this one's very large and white. It also like has that elevation as I was talking about. This one has a completely different type of elevation and margin. And then you will also look at hemolysis of the bacteria. So is the bacteria using up the blood? So this is usually made with either sheep or horse blood, the blood agar plate. So you want to see if the bacteria is consuming this blood, and that is referred to hemolysis. When it is not consuming the blood, we call this gamma hemolysis. And then depending on to what degree it consumes the blood, it is either referred to alpha hemolysis or beta hemolysis. So as you can see, the beta hemolysis is basically this very like white-like structure because it almost completely consumes the blood, whereas alpha hemolysis usually turns this kind of like grayish color. And the best way to see this is to actually lift your plate up and look from underneath it. If the bacteria is consuming the blood, it's vacant in those areas. So if you were to lift up this plate, it would be vacant in this area. There would be no blood agar here. So that is all on bacteria. If you guys have any questions, please let me know. We will now be moving on to the Protista, Kingdom Protista. We're going to be looking at animal-like Protist as well as plant-like Protist.